The next presenter will be Zeraye Mehari Haile. Zeraye is from Ethiopia. He had his BSc studies in plant science from Alamaya University in 2003, and since then he was working as a researcher at the Ethiopian Institute of Agricultural Research at the Werer Center, focusing in crop protection, and since last year he is a master student in our program. Zeraye, please. Click on this.
Thank you, Shuki. Good morning, everybody. My title is Seed Germination and Early Development of Tomato Introgression Lines under Salinity. It was conducted under the supervision of Professor Saranga. To begin with the introduction, I will start with the effect of salinity on plants. As we all know, salinity is an abiotic stress. It affects the growth and productivity of plants. Wow. One of its effects is osmotic, it's low external water potential that will cause stomatal closure, which will reduce the concentration of carbon dioxide inside the cell. That will lead to for photo reduction. That will result in accumulation of reactive oxygen species, causing oxidative stress. The other effect is it makes certain ions unavailable for being uptaken by the plant, like potassium. That is competition for uptake because they have similar ion radius with sodium. It also makes unavailable calcium, and at severe cases, nitrogen and phosphorus. The other effect, due to the accumulation of sodium and chlorine, toxicity may be there in plants. Sodium can accumulate to a toxicity level that it will interfere with ions, some enzymatic activities, and also disrupts membrane fluidity. The chlorine toxicity is a scorching effect. It burns the leaves. Hey. All in all, it reduces the performance of the plants. Lateral shoot growth will be affected, and premature flowering will be there. This is, in general, how salinity affects the plant. But the thing is, where is this salinization problem is where it's focusing. We all know that we are living in a planet where 3/4 of the ecosystem is saline, but it's obvious that we are not depending for productivity of our crops in these 3/4 of the saline ecosystem, but we are mainly depending in the unsaline part, and that is our issue. That part of productive sources, they are increasingly affected by salinity problem. Of course, the effect is mainly concentrated in arid and semi-arid region of our planets. That is the orange color of this map shows you the arid and semi-arid regions in our planet. It's found in northern Central America, in Southern, in Africa, Europe and Asia, also in Australia. In these regions, the land use system, which is majorly affected by salinization, is the agricultural land. At the same time, this region has aquifers which have more saline than the rest of the world. But why is this happening? I mean, naturally, of course, we have 3/4 of our ecosystem is saline, but the secondary salinization is affecting the rest part. That is because this area is experiencing low precipitation throughout the year. That means the salinity in the root zone cannot be leached down. The next one is high surface evaporation. The area is experiencing high temperature, so along with the evaporating water, salt can come to the surface. The other is weathering of native rocks in the area due to the climatic factors. And of course, our contribution to poor cultural practices and forest clearance that affects the physical and chemical property of the soils, which increases its susceptibility to salinity. On top of that, as I mentioned earlier, the aquifers in this area are more saline, and they are also the source of our irrigation. So this adds the salinization problem to be severe. These are the list of countries which use saline irrigation in the world, including Ethiopia and Israel. In addition to this, there is one anticipation that about 50% of the arable lands will be saline by 2050. That means almost we'll be losing our productive lands to salinity. In this area, actually, many crops are grown, of which tomato is one and most economical. That is one reason to consider in the salinity in tomato. The other one, tomato is becoming a model crop nowadays for quantitative traits. We will see the reason in a minute. Okay, to start with, tomato is naturally perennial but cultivated as annual. Tropical plant domesticated in South and Central America. Currently is cultivated in a range of climatic conditions, including in the Arctic circles. Actually, high productivity of this crop is realized in areas having warm climates in the Mediterranean region, southern and western part of the US and Mexico. The leading producer is China, followed by US. Previously, we saw the effect of salinity on crop. Now we will see specifically to tomato in brief. When we characterize most of our cultivated tomato varieties, they are sensitive to moderately sensitive to salinity. Salinity affects us actually tomato from germination to yield reduction. It can cause 70% delay, additional days for tomato seeds to germinate at 100 millimolar sodium chloride salinity, or even 125% more days at 150 millimolar salinity than if we use medium without salt. Wow. It also slows down the shoot growth and of younger seedlings. As compared to freshwater irrigation, we can lose 50% of our yield if we use saline water of about 100 millimolar salinity concentration. In general, seedlings are more sensitive to salinity than matured plants, in a way that a concentration that can kill seedling, a concentration that can kill seedling can be tolerated by matured plant. That is why our focus is on seedling germination and early growth, because if we have varieties having good tolerance at seedling level, then we can guarantee that their productivity will be maintained at the end. Yeah, previously I mentioned that tomato is becoming a model crop to study quantitative traits, including salinity. And the reasons are, of its family, it's one of the most studied one. So we have a wealth of knowledge about the genetics and physiology of it. On top of that, it can be successfully be crossed with its most wild relatives and give rise fertile progenies. This helps us to have to reach itself with QTLs conferring for different type of various stresses. On top of that, there are also many interspecific segregating populations in tomato that are very important for quantitative trait studies. For example, we have salt-sensitive cultivated tomato and salt-tolerant wild-type interspecific segregating populations. In addition to this, we have also introgression lines. We will see what introgression line mean in next slide. About 5 introgression lines so far, and these introgression lines hold segments of exotic genetic materials in the background of cultivated one. They are very good for quantitative trait studies. To define with introgression line, it's a line containing a single introgression from wild tomato genome produced by successive backcross in the domesticated background. How it's done is here presented schematically. We cross elite varieties with wild, and then backcross with the elite variety again. Then we will, using the marker-assisted selection and consecutive backcrosses, we will end up with such lines. And these lines, say for example, if we take introgression line 1-1, it has this segment from the wild type, but the rest from elite. And if we compare for any kind of quantitative trait, this one with it is elite, and if from its parent, and if we found that some difference, then that difference is attributed due to this segment. That is why it's very good. I mean, good lines to deal with quantitative traits. I told you that there are 5 introgression lines so far, among which this is the one introgression library we used for this experiment. It composes Solanum penile accession, this one in the background of a cultivated tomato cultivar M82. This is the most studied IL library for most of the quantitative traits, including salinity stress actually. We are selecting this one. One is available in our lab, and the other, the donor one, is salt tolerant. With this background, the objective of this study was to assess the effect of salinity on germination and seedling development, and to identify the genomic regions conferring salt tolerance at emergence and early development. As the material method, we use 76 introgression lines along with their parental cultivar. 20 seeds were selected and germinated in petri dish with 2 salinity levels. One is 0, having only 3 millimolar calcium chloride, and the other is extra 120 millimolar sodium chloride. We actually reached to we selected this concentration based on our preliminary study. We conducted preliminary study on 4 cultivated varieties and produced a germination curve. Of that germination curve, we were interested on a point where 50% of the germination was reduced, and based on that, we selected this one as a treatment. Why 50%? One thing, we need the seedlings to evaluate after they germinate, and I mean, we need the seedlings, so we don't need to kill all of them by heavy stress, and also having 50% a level which kills 50%, that gives you to identify those performs well. I mean, they can express themselves well in this condition. So those express themselves well can be good to study for further salinity studies. 10 uniformly germinated seedlings were then transferred to pots filled with coarse quartz sand. They were irrigated daily with Hoagland solution, having a 120 millimolar sodium chloride concentration, and without that. The experiment was actually conducted in a growth room, having this humidity 70%, 25 degrees centigrade temperature, and 11 light hours. The experiment was laid in factorial design. Seedlings were uprooted 20 days after transferring for leaf number count, shoot and root lengths measurements. Shoot and roots were oven dried for 72 hours at 60 degrees centigrade. We also did mineral analysis on sodium, chlorine, and potassium. For this, actually, we didn't use all of the lines because it's time-taking, and we selected 15 best-performing lines to analyze for it. In order to extract the solution, we used 0.01 normality nitric acid, and it was shaken for 3 hours by shaker, then centrifuged for 10 minutes, and we used flame photometer for sodium and chlorine analysis, sodium and potassium, and chloride analyzer for chlorine analysis. Of course, we use standard solution to calibrate the standard curve and and to calculate the amount of ions in the sample we used. Result and discussion. Actually, salt stress affected negatively almost all of the growth parameters we considered. This is the first result. Germination percentage and time to 50% germination. This table shows you time to 50% germination. The yellow highlighted ones are those who have those which takes less time to germinate 50% than the M82, and those with green highlight that takes longer time than the control. As you can see, these lines have shorter time to germinate for 50% than the M82. This speed of time actually very important at field condition even, because if you use saline water or even on saline land, there is a crust forming problem, and these are when seeds germinate sooner, then they can bypass the crust formation case, so the stand establishment will be well then. The next highlighted one are those who performed well at relative. When I say relative, their performance under treatment as compared to their performance under control. This one is the germination percentage under control condition, A, and treatment, and relative term. Under control, three lines. By the way, this line, the 0 lines, shows you the performance of M82. We bring the performance of M82 as 0. I mean, we centered every performance toward the performance of M82, then the M82 becomes centered to 0, and every deviation from that is, if it is negative, then lower performance. If it is in the positive axis, it is higher performance than the M82. And under control condition, only 3 of lines have lower germination percentage than the rest, whereas at treatment, IL64, 14, and 72, 925, 741, 913 got significantly higher percentage, germination percentage, than the control in relative term. In addition to this, 925 and 83 has got significant germination than the control. These are their performance were significantly lower than the control. IL14, 64, 72, 741, 925, and 93, they have significantly better germination at salinity, and as well as they germinate earlier. Their 50% germination was earlier than the control under salinity condition. This is the leaf number. As you can see, some of the lines has got lower leaf number even under control, and some of the lines have got higher leaf number under salinity treatments. These are the lines which have got a higher leaf number than M82 under salinity treatment. This, the yellow circle, is about 913. This is so far is superior. I mean, it got higher performance in germination, in early in time to germinate, and in also in the leaf number. Having high leaf number can guarantee you have a high photosynthetic efficiency, of course. And the other parameter we looked for was shoot length. In shoot length, it's only one IL-11-2 had higher shoot length than the control at treatment. And when we consider the relative performance, IL-13, 831, 83, 741, 5144, 14, and 11-2 had higher performance than the M82. Relative to non-stress condition, these lines had performed good than M82, and the underlying one has good performance at germination as well as at time to germinate. 913 still is having a higher performance in every parameters we saw so far. For root lengths, it's only IL-12-11 got higher root lengths than the M82 at treatment, none at relative term. Actually, salinity slows down root growth. It's only 12-11 root was significantly longer than M82. Under stress condition is expected, to, especially for tomato plant. The other parameter we looked at were shoot dry and root dry mass. For IL-44 and IL-11-2 has higher shoot dry mass at treatment level, and IL-813 at relative term, its shoot dry mass was higher than M82. When you come to the root dry mass, IL-11-2 and 12-4 had high root dry mass than M82 under stress condition. 7-2 and 8-3-1 and 9-4 in their relative performance. The other thing we did was shoot mineral content, and I present you here potassium to sodium ratio and chlorine. In control condition, there wasn't any difference between the lines and the control, but for chlorine, some of the lines got lower than the M82 under control condition, and one line has higher 11, that is 11-2 has higher than chlorine. This line, it has also higher sodium to potassium to sodium ratio, chlorine, and sodium concentration also. And in the previous slides also, it's got higher root dry mass and shoot dry mass and higher shoot lengths. By the way, wow, sorry, yeah, having high chlorine content and sodium without reaching their toxicity level can help to modify the osmotic relation. I mean, there are also osmolytes if they are not under toxic concentration, so they will help it to have more solution flow. That may be the reason why this line is performing good, though its percentage of germination performance is very poor. Okay, this is the main part of this study, I mean the mapping, and based on the performance they showed, we tried to map the ILS. In the first chromosome, the segment IL-14, it has high germination percentage. By the way, the black one is for control, red is for treatment, and green is for relative term. And the red and underlined, they are for both relative and treatment. So IL-14, it has association with increased germination, decreased time to 50% germination, and increased shoot layings at 50%, at relative term, I mean. The segment 2-4 is mostly associated with unfavorable traits, reduced shoot layings, dry mass, root layings, both under control and treatment level. In the third chromosome, only one segment was associated with decreased shoot layings that was also even under control condition, whereas on the fourth one, 4-4 has associations with many of the traits: shoot dry mass, shoot layings, germination percentage, and reduced time to germination. 4-3, it is associated with leaf number at treatment. In chromosome 5, most of the associations were not for favorable traits, but on chromosome 6 we have 6-4. It has good germination percentage, lower time for germination, and at treatment level. On chromosome 7, segment 7-41 was associated with reduced time, both at treatment and relative term, and increased shoot layings and increased germination at relative term. In 7-2, increase in germination and leaf number and decreased germination time was associated. In chromosome 8, segment 8-1, it was associated with increased leaf number, increased potassium to sodium ratio, and time to 50% germination. 8-3 and 8-3-1, they were found associated with increased leaf number, shoot layings, potassium to sodium ratio, and high germination in their relative. In chromosome 9, segment 9-1-3, for increased leaf number, decreased time for 50% germination, decreased sodium content, increased potassium to sodium ratio, that is in treatment under stress condition, and these traits for relative. On segment 11, I mean, 11-2 was associated with increased shoot layings and dry mass, root dry mass, sodium and potassium to sodium ratio, increased shoot layings, and increased chlorine at control condition. In chromosome 12, in 12-1-1, it was associated with increased root layings under salinity stress. In general, 154 Q12s were found associated with different traits. Under control condition, 12 loci were associated to be responsible for these traits. Under unfavorable 21, under salinity stress, 32 Q12s were associated with favorable effects under salinity, and 34 with unfavorable condition. In relative term, 39s were found associated with favorable traits, and 16 with unfavorable. In conclusion, germination and seedling development of most of the ILS and M82 were negatively affected by salinity condition. Out of 154 Q12 associated with germination, seedling development, and mineral content, 71 were responsible for favorable effect under salinity and in their relative term. these lines, IL14, 44, 64, 81, 91, and 913, they were superior in germination and for most of growth parameters measured. For IL2, it has lower percentage germination under salinity condition, but the shoot length, shoot and root dry mass, and it has also associated with high sodium, potassium to sodium ratio. Yeah, QTL identified for having positive response under salinity can be used for further salinity stress. Actually, this is the reference I used. Thank you.
Question, Alicia?
Thank you very much. First of all, I have to leave, so I have to ask two questions, and I apologize. The calcium and the sodium chloride combination. Is this aiming the calcium for the ions in the calcium chloride as saline, or is it aiming for a different purpose? What do you know about the combination of calcium and sodium from studies which have been conducted actually here in Israel?
When the calcium concentration is good enough, it protects the root. I mean, the selectivity for potassium will be high under good condition of calcium. That is. At which
plant? I mean, at which crop was the calcium effect actually made very clear, very evident? Can you recall from literature?
No, but maybe tomato.
In cereals, actually, that was a very clear effect, like having 10% calcium or 5% calcium ions compared to sodium chloride. And Professor Kofkafi, which is in our building in the plant sciences, has become actually the world leader in the understanding of the moderation of salinity severe effects by calcium ions. This is number one. Number two, could you tell me why was the calcium included only in the germination and not later on when the plants have been developing?
Okay, on the germination, we want to see only the effect of sodium chloride. That is why we added 3 millimolar of calcium. But later on, the solution we used is Hogland. Calcium is already there. We don't need to add it. The only thing we need to modify that solution is for its salinity. We added an amount of sodium chloride to make a 120 millimolar watering solution.
I see.
Yeah.
Okay, I'll have to leave, so I apologize.
Thank you, Alicia. Who else for questions, please? Dafna? Dafna? There, Janak?
You mentioned earlier that osmotic effect in introduction leads to ROS accumulation.
Yeah.
But as far as I know, I have never encountered any literature that suggests that osmotic effect leads to ROS accumulation. What do you say about that?
Here you mean?
Yes.
You said that you never come across.
Never encountered any experiment in which osmotic stress is implied, but the level of ROS was not induced or like this.
Just simple logic. Osmotic, one of the effects of salinity is osmotic.
Yeah.
That leads to stomatal closure. What stomatal closure will bring in lower concentration of carbon dioxide. There is Rubisco. It's active for oxygen and carbon dioxide. When the oxygen concentration is high than carbon dioxide, it will tend to work with oxygen, right? When it works with oxygen, of course, ROS are always there, but their concentration matters.
Yes.
When there is not enough CO2 concentration, the production of ROS will be higher.
I am not saying that. Before, it was considered the logic is okay. But while doing experiments, they did experiments many times because salinity is the main problem. The experiments are just running everywhere on salinity. But when the experiments are being run, then they never encountered ROS accumulation during this stress. So they consider this as a different, then they took it differently somehow. Though it's still questionable, it's not clear, but they just started thinking that it's a little bit different from ROS accumulation. I don't know why, but if you can explain.
But not being able to identify it doesn't take us to the conclusion that it will not bring, a ROS. But I referred on many literatures, and under salinity stress, ROS are also there. I've also read on literatures.
Okay, but I never encountered the condition that the osmotic stress induces ROS.
No, look, the thing is, for biotechnology assignment, I did on it, and I consulted a lot of literatures, and I found that. That is it.
Okay, that might be your fun, but even I worked specifically on ROS, and I never encountered the situation that the osmotic stress or the salinity stress they itself induces ROS.
No, what I mean is, look, not being able to identify it at one or twice of the trial, it doesn't take us to the conclusion that it will not, you know?
Next, please. Let us not focus on this aspect because it's a marginal thing to this work.
Yeah. You told that the seedling does not tolerate the salinity than the older plants. What's the physiology behind this?
Okay. I mean, when you are matured enough, you are enough to tolerate everything, right? Than being seedling, that is why. I mean, at seedling stage, they are vulnerable for every kind of stress. I think that is common for every plant, I guess. And that is happening here also in tomato. I mean, when it's seedling, yes, it's vulnerable for every kind of stresses.
No, but I was asking about the physiology behind it, yeah.
The physiology behind?
Yeah, maybe some osmotic stress.
Yeah, I mean, they can't adjust their osmotic, the capability to adjust their osmotic stress at seedling stage and at later stage is not the same. I mean, at the later stages, they have a lot of accumulated assimilates, and they can adjust by breaking those hexoses to maintain the water flow. But in this case, they are seedling, so I don't think they have that much accumulations of osmolytes to counterbalance the effect, the stress, I mean.
Okay, one simple question. What's Hoagland solution?
What's Hoagland solution?
Yeah.
Okay, Hoagland solution is a solution containing all nutrients to enable plants to grow without soil. I mean, it will contain every nutrient that enables plants to grow.
Specifically for tomato or
it's general.
It's a standard
nutrient.
Zeraye, not from the scheme, but practically, how do they develop the integration lines?
The introgression lines?
Yeah.
Here?
Yeah, but not, you didn't develop them, right? How, they actually have to cross them with the lead cultivar that it was, I think, M82?
Yes. For us, for our case, the elite variety will be M82, and the wild variety is the penny Lee that is actually resistant for salinity stress. You cross them, you backcross the F1 with the M82, then there will be successive backcrossing.
No, but the part that I don't understand is in the, you have the elite variety, let's say it's M82, and then you have the other one named that it's
penny Lee, okay, Solana penny Lee.
So then you have the hybrids.
Yes, you have the hybrid.
Okay, from the hybrids, how do you go to identify the chromosomes and say, okay, so
okay,
this
for
region
that
from
use marker-assisted selection. Then
yeah.
you will identify on chromosome number 1 at which point the segment is there. I mean, later on you will be having such map. Say, for example, this is chromosome number 1. At this chromosome number 1, this segment is replaced by this one, and it is IL112. You have this line only, having the rest, the background of M82. So any difference that is you observe it will be accounted due to this one, because the rest is the same.
Okay.
You didn't get
After,
me.
since I was involved in the history of developing these lines, I can say that after crossing and producing the backcross 1 was grown, then the progenies, the backcross 1 F2, backcross 1 F3, etc., were grown for several generations. Then the outcoming lines, after they were somewhat genetically stabilized, were taken and scanned with genetic markers. So based on these genetic markers, lines were selected that have single introgression in different parts of the genome. Now, in some cases where the genome was not covered completely, they had to go back to different lines and find lines where they had these introgressions in order to complete the entire set of ILs that gives a full coverage of the Lycopersicum pinelli genome in the Esculentum background.
One question for you, Zeraye. You told that in introduction salinity caused the earlier flowering. How it is physiologically, how it is trigger the earlier flowering in the crops because of salinity?
Because of, okay, Bacteria early flowering, premature flowering is a response of plant for for many of the stresses. I mean, when they sense that they are under stress, they want to finish their life cycle as soon as possible to give a progeny. I think that is the reason why the prematurity will be there, premature flowers.
It's premature or earlier flowering?
That prematurity is earlier. It's the same, right?
No, it means physiologically how suppose sodium can trigger the earlier flowering.
Sodium can trigger earlier.
Suppose sodium, how it help to trigger the earlier flowering?
Sodium can't trigger earlier flowering. Actually.
No, you told that salinity.
Yes, when plants are under stress, I mean, when the conditions are not favorable, that is they have signals. Many signals are produced due to that stress, right? That signals may be to close up its life cycle soon. That may be the case for prematuring. I mean, earlier flowering and the like.
I have a question. From your conclusion, you said that there is 154 loci that are related to salt stress. Is that the conclusion?
Yeah.
And so is there like a difference contribution to each one of these loci? I mean, if you want to breed for salinity, for more salinity resistant tomato variety,
Yeah.
so like from your experience, do you know which loci you need to aim that you want to include in this? I mean, maybe you said it, but I didn't understand.
Look, yeah, every traits are, I mean, they are controlled by different locuses. That's why, and one trait can contribute for many one locus can contribute for many traits too. Here you can see that for germination percentage we have three lines. They are superior than the M82. For leaf number 6 lines. Maybe this say 913 is associated with all these favorable traits under salinity. So if there is any breeding program interested in specific
So this one would be more interesting to include than
there are more of this one actually.
Mm.
Yeah, so they can be.
Okay.
My question is, the lines were not consistent for germination and dash to germination percentage, and also for some of the lines had superior result for germination percentage and dash to 50% germination, but in terms of biomass accumulation, other lines, I think particularly this IL9-3 it was superior in terms of germination, but it was not superior in biomass accumulation, I think.
Yes.
So which trait is important to map the QTLs, germination or biomass accumulation?
No, we are mapping the QTLs according to their effect on those traits we observed. I mean, it's not a must for one line to perform everywhere. I mean, in every trait, because every trait is differently controlled by different. They are differently controlled by different locuses, right? I mean, a locus which is responsible for high germination under stress cannot be responsible to have high leaf number under stress. Cannot be responsible to accumulate high biomass. I mean, it's different. That is why salinity stress are quantitative traits. Stress are quantitative traits. Many traits, many locus involves. I think if I got you.
One more question. Yes. How many replicates you have? 5 replicates. And in every you build the QTL mapping?
No, the QTL
based
is the last. The QTL is based on this performance. I mean the statistics I showed you on tables and the graphs. This is based on this one. Say IL64. These lines, they have significantly lower time to germinate to reach 50% germination under salinity. That means it shows that they have something to do with speed of germination.
Okay, thank you.
Did you find some QTL that affects positively in some trait in salinity under salinity stress and negatively in another trait during salinity stress?
Okay, important in one, yes.
Yeah. At the same time?
It means contradicting effect. Let's say enhancing germination but reducing leaf number.
Yes, like this.
I think there are. Here increase leaf number, decrease shoot length.
Okay.
There will be, yes, because I mean there are different traits, different characteristics. They can be affected differently by one locus.
Okay, thank you very much.